All right, we will continue our discussion of the cell, picking up with parts of the cell and the cytoplasm. So recall from last time that we talked about the different types of transport across the cell membrane. Now we'll just zero in and talk about different parts of the cell, talking about their structures. So the first slide here talks about the cytoplasm. The cytoplasm includes the fluid material inside the cell. You can almost think of it as a soft set jello, like a jello that's not quite ready to be taken out of the refrigerator and served. So it has a little bit of consistency to it. It helps suspend the organelles. And the cytoplasm also consists of a cytoskeleton, which is a network of microtubules. It kind of serves as a scaffolding within the cell as well, and that gives the cell its shape. Organelles within the cytoplasm, there are several that we're going to talk about next. All of these are membrane bound. They have a specific function in relation to the rest of the cell, and we'll talk about those functions as we proceed through our discussion of the different organelles. So when we talk about the nucleus, a lot of textbooks will say that the nucleus is the control center of the cell. That's true to a point. However, the nucleus, it consists of this membrane here with pores in it, and inside of it is a space which contains DNA. And it's the DNA that really controls the processes of the cell, because the DNA contains the information for making protein. And protein is essentially one of the most important molecules of the cell, because a majority of our structure is protein, and many of the enzymes that we produce to drive chemical reactions and metabolism in the body, those also are proteins. So the nucleus is simply just a space which contains DNA and another organelle called the nucleolus. So you can see these kind of purple hazy structures here. That's DNA in the form of chromatin, and in lab we talked about how chromatin is a loosely arranged form of DNA. So essentially the nucleus is just a space in the cell surrounded by a membrane called the nuclear membrane, which contains DNA and this smaller organelle called the nucleolus. The function of the nucleolus is to make ribosomes, and we'll talk about those in a little bit. So the nuclear envelope is very important to contain this DNA, because DNA never leaves the nucleus in our cells. It remains housed within the nucleus. And again, the nucleolus is in charge of making ribosomes. Here we can see the next organelle we're going to talk about, and that's the rough ER. So here we can see the rough ER, that its membrane is continuous with the nuclear membrane. So we have the space here that contains the DNA. This is the nuclear membrane shown in purple with the pores in it. And continuous with that is the membrane of the rough ER. And what makes the rough ER rough are these small pink circles you can see, which are ribosomes. So these are tiny organelles whose job is to make protein. So they take the information from DNA with the help of RNA to make proteins. So all these little red dots we see here are ribosomes. And they are attached to this membrane called the rough ER, which we'll talk about next. So we can find ribosomes attached to the rough ER on its surface, or we can find ribosomes free floating in the cytoplasm. Either way, the function of ribosomes is to make protein. So any cell that is in charge of making enzymes, for example, like the pancreas, some of the cells of the pancreas are in charge of making digestive enzymes. Therefore, we would expect to see a lot of rough ER and a lot of ribosomes in those cells, because of their primary function being to make enzymes, which are proteins. So here we can see the endoplasmic reticulum, and it's zoomed in here to show you just how that membrane of the nucleus becomes the rough ER. So if it's a substance that leaves the nucleus, for example, DNA information being copied to RNA, can move out here into the rough ER, where those proteins are made on the surface via these ribosomes, and then those proteins channel through the rough ER and are modified chemically. So the function of the rough ER is to take those proteins that are produced on the surface by the ribosomes and modify their structure. So the rough ER modifies the structure of proteins. Now the smooth ER does not have any ribosomes attached. The smooth ER's job is to manufacture fats or lipids. Some of the hormones, for example, that we need in our body, like estrogen, testosterone, progesterone, cholesterol, those are fatty molecules, and it would be the smooth ER in the cells of those endocrine organs that would make that hormone, those fatty hormones. So it's the smooth ER. So it looks just like the rough ER. However, we're going to see that without ribosomes attached. So if we look at our model here, looking for the smooth ER, we can see here there's no ribosomes attached to the surface here. That would be an example of the smooth ER. Okay, moving along then, the next membranous organelle involved with protein is the Golgi apparatus. So the Golgi apparatus is a network of membranes just like the rough ER. However, it does not have ribosomes attached to it, and it looks a little different compared to the smooth ER in that the membranes are isolated in one area of the cell and they're very much flattened. You can see how it looks almost pinched together here in the middle. These membranes are very flattened in the center, and that's characteristic of the Golgi apparatus. If you look in your textbook on page 69, you can see the Golgi apparatus, how that looks very different from the rough and smooth ER. They typically use the same color in a model or a diagram for both the smooth and rough ER, and they'll use a completely different color for the Golgi apparatus. So let that be a guide to you also, that that's a very separate and unique organelle. So the job of the Golgi is to modify those proteins a little bit more structurally and also to package them into these vesicles. So here is just simply a piece of the membrane of the Golgi apparatus that has been pinched off and contains a protein that the Golgi, working with the rough ER and the ribosomes, produced and modified. So this might be a digestive enzyme, for example, coming from a cell of the pancreas to be secreted into the small intestine to help us digest different components of our food, like proteins, fats, carbohydrates. Here we can see how the rough ER with the ribosomes attached to the surface make this chain of a protein. That's what's shown in blue here. And that protein is modified structurally by the rough ER. It's packaged into a vesicle. Here it's expanded down here, so that vesicle is pinched off. It's taken into the Golgi apparatus. The Golgi apparatus further modifies it, then sends it outside the cell to be exported. And I have a little animation here that we can watch that process. Let's move to the internet. So here's the nuclear membrane. We can see there's pores in the nuclear membrane. Here's the rough ER with ribosomes attached to it. And we can watch, this is DNA here in the center. So as this process continues, we can see the DNA is unwound. Messenger RNA comes in, copies the information for making a protein, takes it out into the cytoplasm. It attaches to some ribosomes. And these ribosomes will read the information from messenger RNA to start making a protein. The little green strands you see here, those are little segments of protein that are being assembled by the ribosomes. Those ribosomes are attached to the rough ER, and those proteins go through the network of membranes of the rough ER, and then they're exported by exocytosis. So here's the rough ER again. Here's the proteins that have been produced by the rough ER, or by the ribosomes on the surface of the rough ER. Then the rough ER modifies those proteins, and then we can see how they are packaged into little vesicles, and they're going to be taken in by the next organelle, which is the, do you know? The Golgi apparatus. If you said that, that's correct. So endocytosis, taking those proteins into the Golgi. It modifies those proteins chemically again and exports them by exocytosis to the plasma membrane, which would be right here. So we'll see those proteins go to the surface of the plasma membrane. Again, exocytosis, it enters into the fluid outside the cell, and that protein is off and on its way to do its job. So that's how the rough ER, the ribosomes, and the Golgi apparatus work together to manufacture proteins. Lysosomes are simply packaged digestive enzymes. When you see the prefix lyse, think of slice and dice or break down. And that's what the lysosomes do, is they break down different structures within the cell. These might be digestive enzymes that would destroy, say, a bacteria that has invaded the body. It might be a digestive enzyme to break down a protein or a lipid in the small intestine or the stomach in the case of a protein. So again, these are any membrane-bound digestive enzyme is called a lysosome. Your textbook shows that very well on page 70, shows how we have lysosomes being produced by the Golgi apparatus as it packages its proteins that it reassembled into these vesicles. So page 70 has some nice pictures in your textbook showing lysosomes. Mitochondria are kidney bean shaped structures that are scattered throughout the cell. There's definitely more than one of these, and their job is to take the glucose that we bring in via our food, which is absorbed into our cells, and in the presence of oxygen makes ATP. We talked last time how ATP is the energy currency of our cell. It's what our cells directly use to drive energy requiring processes. So the mitochondria is in charge of making that ATP. So when we think of the mitochondria, think of them as the energy powerhouses of the cell. Any cell that needs lots of energy for a specific function, we would expect to see a lot of mitochondria. For example, heart muscle cells. The heart is constantly pumping to drive blood throughout our body, and it does that for years and years and years. As a result, we would expect to see a lot of mitochondria in heart muscle cells. And in an athlete who's been training for an extended period of time, our body responds to the demands placed upon it. So a person that's been training for a long time, over time, through the benefits of training, muscle cells in athletes will develop and produce more mitochondria to produce the energy that is being demanded from those muscles. Cilia are extensions of the plasma membrane of cells that waft back and forth, or waft in one direction depending on what the purpose is, to move substances over the surface. For example, lining our respiratory tract, we have cilia that helps to take mucus with trapped debris and move it toward the digestive tract. For example, every now and then throughout the day you'll feel the urge to clear your throat. When you clear your throat, you're taking that mucus that is being moved upward in your throat by your cilia, and you're pushing that further upward to be swallowed down your esophagus and to direct that mucus into the digestive tract. The same thing happens in the back of our nasal passages. The cilia beat backwards to beat the mucus toward the back of our throat, and every now and then you feel the urge to swallow to move that mucus that's collecting in the back of your nasal cavity toward the digestive tract. So cilia are short, numerous extensions on the surface of a cell, and the job of cilia are to move substances over the surface. Flagella a flagellum is a single whip-like structure that helps to propel or move the cell itself. And we only see one flagellum in the human body, and that's with the male sperm. So the job of that flagellum is to propel that sperm, but specifically the head, which contains the nucleus full of DNA for making a baby. It propels that cell up the female reproductive tract to the fallopian tube to find the egg and unite that nucleus of the sperm with the egg. So that is the purpose of the flagellum, is to propel the cell. Microvilli, those do not move. They are extensions of the plasma membrane, but they do not move. The only job of the microvilli are to increase the surface area for absorbing substances. This we find in the digestive tract. If we had a single columnar cell, it would be not advantageous for absorption purposes if we had one solid membrane across the surface here. This distance from here to here for absorption is not very long, but if I add in the distance of each microvilli going up and down, that's quite a considerable difference in size for surface area absorption. So microvilli do not move. The only thing they do is they increase the area for surface absorption. So it's like looking at the fingers on your hand. If you just held your hand out straight, each finger would represent a single microvilli. and your hand would represent the cell. So rather than having a membrane just go straight across with no fingers, these finger-like extensions again increase surface area for substances to be absorbed, and we find these again along the digestive tract, particularly starting with the stomach down to the large intestine. So that concludes our discussion of the cell and mitosis. Be sure that you're looking at your syllabus and you're taking the lecture exam when you're ready. And our next lecture exam on this material will be the fourth week of class. So that check your calendar. Friday by 5 p.m. is when this material, along with tissues and skin, will be due for testing purposes. So you have yet to watch the tissue and skin lecture video before you'll be ready to test on cell, tissues, and skin. Thank you.